maybe what happened? Man, I don't know what. I wonder what's in this one. Okay. Oh, my stomach's already full. Anyways, let's do immune system. Let's do. I don't know if we have time for this one, to be honest. We might. Let's do this one, and then let's do. I want to do embryology a little bit. We have quite a lot to do today, and I just want to hit this video for respiration, just to make sure it's good, and then go over viruses and bacteria, and we'll hit his biochemistry a little bit later. Like when we get here, I want to do some kind of academy on the rest of the bio, like the organ systems. I want to finish the organ systems. Really need to finish. Actually, I'll just break it here, actually, so that I know to stop here, because the rest of the stuff is a little bit. Well, same as this one, but the rest of it is pretty much review. Excuse me. These are some good videos, huh? Yes, sir. Okay. Continue. How does it know? How does it know to release it? Wow, amazing. Let's take a break. Just take like a 10 minute or something. We'll be back. I'm not sure yet. I think that's something with dynamics. All right, let's take a break. Let's take a 10 minute. Yeah, we have a couple more videos I want to watch. Is this one? This one? Yeah, I already went over this. Anyways, 10 minutes. Let's continue. I have to Anki today. Okay, I'll be back. Just put this down or something.
gotta go. I gotta go. I gotta go. I gotta go study. My life depends on this test. My life depends on this test. All right. Oh, wait. I lost it. That's fine.
Maybe I should get on Mozilla. I really don't want to have any more ads. Freaking ads, man. Wasting my freaking time. I think there's no ads on Mozilla, right? Is there ads? Mozilla Firefox. I thought it already came built in. Oh, really? I thought it did. That's strange.
Hey guys, how's it going? Today, in this video, we're going to do extension. Alright, let's navigate to extensions and see all.
Oh, they say just get you black origin. I don't have it. Yeah, I already have it actually. I already have it. System playlist.
Unlike
maybe it's better. I think it's better we switch because I don't want to keep having these problems. 534. Oh wait.
Whenever a pathogen invades our body, it brings along
That's fine. Let's exit out this one then. There we go. Something like that, I think. Fine. Yeah, I think so. Okay.
every pathogen invades our
We don't have
body.
to deal with any more ads. Yeah. Yeah.
It brings along its own proteins, its own nucleic acids, and its own polysaccharides, known as the consequence of amino acids for them to actually bind onto different types of antigens. Now, on top of that, it's these variable regions that contain these clefts, that contain these regions that actually bind onto our antigen. So these are known as the antigen binding sites. Now, the region of the antigen that the antibody is actually that binds onto the antigen binding sites of the antibody is known as the antigenic determinant, or simply the epitope. So if we look at the following diagram, we have our antibody that contains the constant region and it contains the variable region. And it's the variable region that contains the antigenic binding sites, as shown, that can actually bind onto the epitope of our antigen. So let's suppose we have the antigens floating around in our blood or in our lymph or in our tissue. Now, when this specific antibody that was built by the plasma cell approaches these specific antigens for which it was built for in the first place, we have a binding process taking place. And the epitope basically binds onto the antigen binding site of this antibody, as shown in the following diagram. And then we form our antibody antigen complex, which then elicits some type of defensive response, as we'll discuss in just a moment. Now, what about the constant regions? So basically, earlier we said that we have five different classes or five different types of antibodies. Now, each class of antibody has the same exact constant region, but these constant regions do change when we go from one type to a different type. In fact, these constant regions determine the type of class that we're dealing with, and they actually determine the mechanism that our antibody uses to basically fight off infections and fight off these pathogenic invasions. And on top of that, it's these constant regions, the bottom portion of these constant regions, that actually bind onto the cell membranes of immune cells and other cells of our body. So now that we know what the structure of the antibody looks like, let's discuss what the mechanism is by which the antibody actually elicits a defensive response by our immune system. So antibodies can either be found bound onto the membrane of immune cells, or they can float around as free antibodies within the blood or lymph of our body. And either way, when the antibody locates and binds onto that specific antigen for which it was actually built for, they form the antibody antigen complex. And once the complex forms, it elicits some type of response that begins a defensive mechanism to kill off that particular pathogen. Now, what type of mechanism do we actually have? Well, we have three important types of mechanisms that we're going to focus on in this lecture. Let's begin with mechanism number one. So in this case, what happens is when our antibody binds onto the antigen, it basically inactivates that antigen. So let's take a look at the following case. Let's suppose we have a virus that basically infects our body. So the virus is swimming around in our tissue. Now, our antibodies, which are specific to the antigens found on this virus, when they locate the virus, they will bind onto those antigens. And what happens is, because the virus uses these antigens to actually bind onto the cells of our body, because the antibody actually blocks these antigens, it also blocks and prevents the virus from actually binding onto our healthy cells. And that means the viral agent cannot actually infect our healthy cells. And so this is the process by which, once the antibody antigen complex forms, it essentially inactivates that particular pathogen. Now, let's move on to number two. In number two, let's suppose we have some type of cell that was infected by our viral agent. Now, once that cell is infected, that infected cell releases some type of pathogenic antigen and places it onto the MHC class 1 complex. So remember, MHC is the major histocompatibility complex that our cells use to basically differentiate healthy cells from our infected cells. And once the pathogenic antigen is found on the MHC class 1 complex, some type of white blood cell, a macrophage, a neutrophil, or our cytotoxic T cell, a special type of T lymphocyte, can basically approach this infected cell. And if it contains the specific type of antibody that reflects this specific type of antigen, they can basically bind, and then the cytotoxic T cell can release powerful proteins that ultimately poke holes in this cell and lyse the cell, killing that cell off. And then a macrophage can swim by and pick up the remaining debris from that infected cell. So mechanism number three, we can also actually have a process known as agglutination. And what agglutination is, when we form the antibody antigen complex, many of these complexes can actually aggregate to form an insoluble complex. And this insoluble complex also inactivates our pathogen. And then that entire insoluble complex can be picked up by the macrophage, and basically that macrophage can break down that pathogenic agent. So earlier we said there are 5 different types, 5 different classes of immunoglobulins. We have immunoglobulin D, immunoglobulin A, immunoglobulin M, G, as well as E. So the major difference between these 5 different types of classes of antibodies is the sequence of amino acids in these constant regions. And each one of these different classes of antibodies functions in its own way to basically defend our body from these pathogenic invasions. So let's very quickly look at some major distinctions between these 5 classes. So let's begin with IgG, immunoglobulin G. So immunoglobulin G is actually the most common type of antibody that is found inside our blood. In fact, 75% of the antibodies found inside our blood are the immunoglobulin G types. So what these antibodies do is basically what we described earlier. This was an example of the immunoglobulin G. They basically float around, they swim around our blood and lymph, and they locate those special types of antigens found on our pathogens. Now, let's move on to IgM, immunoglobulin M. So these are found on B lymphocytes, on the membrane of B lymphocytes, and they're highly effective against killing off our viral agents. Now, IgE are those antibodies involved mostly in allergic reactions, and we'll talk about these in much more detail when we'll discuss what allergies actually are. Now, what about IgD? Well, immunoglobulin D are those antigens that are basically also found on the B lymphocytes, and they play a role in actually creating our antibodies. So they help us differentiate our B lymphocytes into plasma cells, and those plasma cells ultimately produce those antibodies that are essentially complementary to the antigens that bind onto these immunoglobulin D types. And finally, we have immunoglobulin A. So immunoglobulin A are those antibodies that are found inside our nasal cavities, inside our air passageways, so the trachea, the bronchioles, as well as the bronchi. And they're also found in our gut, in our digestive tract. They're found in the breast milk. They are found in the mucous membrane along our air passageways. They're also found in our tears as well as in our saliva. So these are the 5 different types of immunoglobulins, antibodies, that are found inside our body, and all of them have this same exact shape. Now, these 5 different classes, of course, have 5 different types of sequences within our heavy chains, within our constant regions, because it's the constant region that determines the type of antibody that we are actually dealing with. To gain a better understanding as to how our
Let's go. Oh, I should have just gone to Firefox a long time ago. Oh, that's great. That's perfect.
immune system actually works, let's take a look at the following. To gain a better understanding as to how our immune system actually works, let's take a look at the following diagram, at the following illustration. So let's begin on this side. So let's suppose some type of infection actually took place, and let's say that a bacterial cell got into our tissue. Now, once the bacterial cell is inside the tissue of our body, infection begins, and the innate immune system is the first one to actually react. And what happens is special cells, immune cells, known as mast cells and basophils, begin to release chemicals such as histamine. And what that does is dilates our blood vessels, and it brings specialized phagocytic immune cells, known as macrophages, to the site of infection. So inside, to gain a better understanding, as to how our immune system actually works, let's take a look at the following diagram, at the following illustration. So let's begin on this side. So let's suppose some type of infection actually took place, and let's say that a bacterial cell got into our tissue. Now, once the bacterial cell is inside the tissue of our body, infection begins, and the innate immune system is the first one to actually react. And what happens is special cells, immune cells, known as mast cells and basophils, begin to release chemicals such as histamine. And what that does is dilates our blood vessels, and it brings specialized phagocytic immune cells, known as macrophages, to the site of infection. So this is what a macrophage actually looks like. So we have different types of organelles, for example, a nucleus, and we also have these digestive lysosomes, these tiny vesicles that carry digestive enzymes as well as an acidic environment. Now, what the macrophage does is it engulfs our pathogen, in this case, our bacteria, and it creates this vacuole within which we have that bacterial cell. And some of these lysosomes fuse with this phagosome, and now this phagosome contains these digestive enzymes and acidic environment as well as the bacterial cell. And inside the phagosome, we begin the process of the breakdown and digestion of that bacteria. So the macrophage essentially engulfs that bacterial cell and begins the process of digestion. Now, the macrophage is actually a type of cell we call APC, or antigen presenting cell. And what that means is after it kills off and breaks down that cell, it takes one of the antigens, one of the proteins or nucleic acids or polysaccharides from that bacteria, the proteins or nucleic acids or polysaccharides from that bacteria, and it places it onto the surface of that cell. But it places it onto a special type of complex of proteins known as MHC, which we spoke about previously. So recall that MHC stands for the major histocompatibility complex. And this is a class 2 complex because it's used to actually communicate with other leukocytes, as we'll see in just a moment. So this antigen presenting cell, our macrophage, creates the MHC class 2 complex. It places it on the surface, and it takes an antigen, which is shown in red, and it places it onto this region on the MHC class 2 complex. Now, what happens next is another type of leukocyte, another type of white blood cell can now interact and communicate with the macrophage as a result of the presence of the MHC class 2 complex along with the antigen shown in red. And a special type of T cell known as an inactivated helper T cell, which is a CD4 plus helper T cell, begins to approach this MHC class 2 antigen. Now, what do we mean by CD4? Well, CD4 is simply a special type of glycoprotein that is located on the membrane of T lymphocytes, and specifically on the membrane of this inactivated helper T cell. And what this CD4 glycoprotein does is it actually binds and interacts with MHC class 2. We'll see that there's another type of CD glycoprotein we call CDA glycoprotein, and that glycoprotein only interacts and binds with MHC class 1 complexes, as we'll see in just a moment. So we have an inactivated helper T cell, a type of lymphocyte, T lymphocyte, that can now go on and bind to this MHC class 2 complex that contains that antigen. So we have the binding process takes place. And when they bind and form this entire complex, the macrophage begins to release a special chemical known as interleukin 1. And the interleukin 1 essentially goes on to and interacts with this inactivated helper T cell. And what that does is it activates that helper T cell. Now, once it's activated, it basically releases from this macrophage, and it moves on and it finds a B lymphocyte. Now, remember, B lymphocytes are part of our adaptive immune system, just like these helper T cells are. But the B lymphocytes are part of the humoral immunity of our adaptive system, while our T lymphocytes and our helper T cells are part of the cell-mediated immunity. So we see that the humoral immunity and the cell-mediated immunity interact with one another to carry out the same exact function to basically kill off these pathogens and kill off the infected cells of our body. So once we activate that helper T cell, it basically goes on and finds a B lymphocyte that contains the MHC class 2 and class 2 complex, along with that same antigen that was found on this macrophage. And they also bind and they also form a complex. And once they form a complex, these two cells begin to release these special chemicals known as cytokines. And what these cytokines do is they basically induce this T cell, the helper T cell, to begin to clone itself via the process of mitosis. So mitosis of the cell takes place, and we form many of these cloned helper T cells. And these cloned helper T cells begin to release another group of cytokines, and these cytokines go on, and then these cytokines induce these B lymphocytes and T lymphocyte cells to basically mature and differentiate into even more specialized cells. So these B lymphocytes basically produce plasma cells as well as memory cells, while the T cell, when it reacts with the cytokines, it produces cytotoxic T cells, also known as killer T cells. And these cytotoxic T cells contain the glycoprotein CD8, and we'll see exactly what that means in just a moment. So let's take a look at these plasma cells. So these plasma cells have very extensive endoplasmic reticulum, as seen in the following diagram. And that's because these plasma cells, their function is to basically form antibodies. So remember, the plasma cells and the memory B cells are part of the humoral immunity. They're part of the antibody-mediated immunity. And what that means is they're involved with forming and storing antibodies. So the plasma cells form antibodies that are complementary to these specific antigens that we spoke about earlier, shown in red. So these antibodies are complementary to the antigens found on this bacterial cell. And these plasma cells form a bunch of these antibodies that can now move around our blood system and lymph system, and they can bind to the antigens of this bacterial cell. Now, memory B cells, instead of forming these antibodies, they essentially store these antibodies within the cell. And they can essentially react with a bacterial cell of the same type that reinfects our body at some later time. So it's the memory B cells that essentially allow our immune system to be adaptive. It allows the adaptive immune system to actually learn from these pathogens that infect our cells of the body. And finally, our T cell differentiates into the cytotoxic T cell, and this contains the CD8 glycoprotein on the membrane. Now, the CD8 glycoprotein basically is responsible for binding to the MHC class 1, the major histocompatibility complex class 1. And this is found on our infected cells of the body. And that's exactly why the cytotoxic T cells are those immune cells of our body that are responsible for seeking out and locating the infected cells of our body. And once they bind to those infected cells, they basically release special powerful proteins that are capable of digesting and breaking down the membrane of those cells, lysing those cells, and killing those cells. And that's exactly why the cell-mediated immunity, the immune system that deals with our T lymphocytes, is responsible for finding and killing off infected cells, while the humoral immune system is responsible for killing off these bacterial cells, pathogens, toxins, parasites, and so forth that infect our body. So this is a general overview, a very simplified overview of how our immune system actually works. All pathogenic antigens that eventually make their way into the tissues of our body must be presented to T lymphocytes, the cells of our cell-mediated immunity. The problem is these T lymphocytes cannot actually directly bind onto these antigens. What must happen before these T lymphocytes interact with the antigen is a special type of immune cell known as the antigen-presenting cell must actually engulf our antigen. And once it engulfs the antigen, it takes that antigen and it places it onto a special protein complex found on the membrane of the antigen-presenting cell. And this protein complex is known as the major histocompatibility complex class 2, or simply MHC class 2. And only when the antigen is bound to this MHC class 2 complex can the lymphocyte actually see and interact with the antigen and bind onto this antigen. And once it binds, only then can it begin some type of defensive mechanism, as we'll see in just a moment. So the three types of antigen-presenting cells that we're going to focus on in this lecture are macrophages, dendritic cells, as well as B lymphocytes, the cells of our humoral immunity system. So let's begin with macrophages. So let's suppose we have some type of infection taking place. Let's say a bacterial cell infects our tissue. Now, as soon as that infection takes place, the innate immune system will try to prevent that infection from spreading. It will try to localize that infection via the process of inflammation. And what inflammation does is it dilates our blood vessels. It brings more blood to that area, and that brings specialized antigen-presenting cells known as macrophages. Now, what do macrophages actually do? Well, macrophages engulf pathogens. Let's suppose we have the bacterial cell as shown in red, and this is our macrophage, and the macrophage engulfs that particular bacterial cell. Now, once that bacterial cell is inside the macrophage forms the phagosome, which contains lysosomes, and the lysosomes contain digestive proteolytic enzymes that break down and digest that pathogen. Now, when digestion takes place, what the macrophage does is, because it's an antigen presenting cell, it begins to build the MHC class II complex, and it takes that complex and places it on the membrane of the macrophage, and then it takes a small peptide, the antigen from that bacterial cell, and it places it on that MHC class II complex. So we have this antigen MHC class II complex that forms. Now, as soon as this actually takes place, we have a special type of T lymphocyte whose membrane contains a special glycoprotein known as CD4. Only T lymphocytes that contain the CD4 glycoprotein can actually go on and bind onto the antigen MHC class II complex. So for example, we have some type of inactivated helper T cell that contains the CD4 glycoprotein on the membrane, goes on and binds onto this complex of the macrophage. And once this binding process takes place, that ultimately activates the helper T cell, and that also releases many different types of chemicals involved in our immune system, and that activates T cells. For example, it produces cytotoxic T cells. It also activates our B cells into plasma cells and memory B cells, and that begins the process of defending and protecting our body from these pathogens. Now, let's move on to the second type of antigen-presenting cell, known as a dendritic cell. Now, basically, all these different types of antigen-presenting cells do the same exact thing. They basically engulf our antigen, and then they present that antigen on this special type of protein membrane complex known as the major histocompatibility complex class II. So what exactly is a dendritic cell? Well, a dendritic cell is a specialized type of immune cell that is found in those areas of our body that are close to the outside environment. And that includes places, tissues like our skin, our gut, our stomach, and our digestive tract, as well as our lungs, so the air passageways of our lungs, like the trachea, the bronchi, and our bronchioles. So this is what a dendritic cell actually looks like. It has these projections, these dendrites. So let's suppose some type of foreign antigen is found inside our tissue, and it comes from, let's say, some type of bacterial cell, the same cell that we spoke of earlier. Now, what this dendritic cell does is it basically phagocytizes this antigen. It basically engulfs it, and the lysosomes break it down, and a small part of it is then expressed. It's then displayed on this MHC class II complex that is formed by the cell, as shown in the following diagram. Now, the dendritic cell is motile, and what that means is it can then move into the spleen or the lymph nodes of our immune system, and it basically begins to interact with some type of T lymphocyte that, once again, has the CD4 glycoprotein, the proper T cell receptor found on the membrane of that T lymphocyte. And when these two cells interact, once again, it elicits some type of defensive immune response. Now, let's move on to our B lymphocytes. So recall that B lymphocytes are those immune cells of our humoral immunity, the antibody-mediated immunity. So B lymphocytes can also engulf and take in these antigens that come from different types of pathogens found inside our tissue. So B lymphocytes are part of the humoral immunity. When they approach foreign antigens floating around in our tissue, they bind to them via special receptors, and then they undergo receptor-mediated endocytosis, and that takes in that antigen into the cell. And once that happens, these B lymphocytes build the MHC class II complex, and they place that antigen onto that complex, and only then can some type of T cell, T lymphocyte with this CD4 glycoprotein bind onto this MHC class II complex that contains our antigen. And once this binding takes place, that ultimately stimulates this B lymphocyte to undergo mitosis, divide many times, and it basically differentiates into two different types of cells: the plasma cells that basically produce the antibodies found inside our blood, swimming around our blood and lymph system, and they also produce those memory B cells that are involved if reinfection ever actually takes place. So once again, we can conclude that the antigen-presenting cell is a type of cell of our body that allows these T lymphocytes to actually interact with the antigens. What these cells do is they engulf these antigens and then they build these special protein complexes, and then they take that antigen and place it on that protein complex, and only then can our T lymphocyte that contains the CD4 glycoprotein receptor actually interact with this antigen MHC class II complex and create some type of mechanism, some type of defensive immune response. B lymphocytes, or simply B cells, are the white blood cells that make up the humoral immunity of our immune system. And what that means is it's the B lymphocytes that produce the antibodies and also store the antibodies in specialized cells in case reinfection ever takes place.
Isn't it the same thing? Oh,
The
I think
question-
it's going over a little bit more in depth here. Alpha, beta T cells, gamma, delta T cells. Allergens and allergic reactions. Immunosurveillance in cancer cells. That is it. That's all 6. We'll take a break here. Let's take a 10-minute. I still got more to go, but it's fine. $14 times 15. 210 divided by 60. 3.5. Dang. That's not even. Alrighty. Yeah, let me take a 10 minute real quick. Another one.
mechanism of action of B lymphocytes. B lymphocytes, or simply B cells, are the white blood cells that make up the humoral immunity of our immune system. And what that means is it's the B lymphocytes that produce the antibodies and also store the antibodies in specialized cells in case the infection ever takes place. The question is, what exactly is the mechanism of action of B lymphocytes? How exactly do B lymphocytes engage pathogens? And how exactly do B lymphocytes protect and defend the cells of our body from these pathogenic invasions? So let's begin by discussing what the structure of a B lymphocyte is. So here we have a B lymphocyte, and notice that the B lymphocyte contains these receptors on its membrane, and they're known as B cell receptors. So let's suppose we have a pathogen that makes their way into our body, and that pathogen, one way or another, will release antigens. Now, these antigens can either float around our blood, our lymph, or our tissue, or the antigens can be picked up by some other type of white blood cell, for example, a dendritic cell or a macrophage. Now, let's suppose our antigen is picked up by the macrophage. What the macrophage will do is it will display that antigen bound to a special type of protein complex on the membrane of the macrophage. And the protein complex is known as the major histocompatibility class 2 complex. And this is shown in this diagram. Now, anytime we have an antigen in our body, either actually floating around in our body or bound to some other type of leukocyte, we're going to have a specific B lymphocyte that has the B cell receptors that can bind to that particular and that specific pathogenic antigen. So the B lymphocyte can either pick up these antigens found floating around in our system, or it can actually also grab the antigen off of our macrophage or some other type of leukocyte. So the entire point of a B lymphocyte is to collect these antigens found inside our body, and these antigens came from some type of pathogen, for example, a bacterial cell. The next question is, what happens once our antigen is bound onto the B cell receptor of our B cell? Well, a process known as cell-mediated endocytosis begins to take place. And what this process does is it invaginates our cell membrane, and it forms this vesicle that is brought into the cytoplasm of our cell, as shown in this diagram. So the antigen shown in red binds onto the cell receptor of the B cell, and then that initiates the cell-mediated endocytosis process, and that ultimately forms that internal vesicle, and now the antigen is found inside this vesicle of our cytoplasm. Now, the cell also has lysosomes, and these lysosomes are shown in green, and they can fuse with this vesicle, and these lysosomes release digestive enzymes that can proteolytically break down our antigen. So the antigen begins to break down, and a small portion of that antigen, known as the antigenic determinant or the epitope, is taken and placed on the same type of protein complex that was found on the macrophage, MHC class 2, major histocompatibility complex class 2. So we have the antigen, we have our MHC class 2 complex on our B cell. What happens next? Well, what happens next is, for any type of mechanism, for any type of defensive mechanism to actually take place, our B cell must interact with our T cell, our T lymphocyte. So a special type of T lymphocyte known as the helper T cell that contains the complementary T cell receptor, complementary to this particular section of our antigen and MHC class 2 complex. And also the helper T cell must have the CD4 glycoprotein to actually be able to bind, interact with the section of our B cell. So here we have the B lymphocyte that essentially contains the antigen bound to the MHC class 2 complex. So if we have a nearby helper T cell that contains not only the complementary T cell receptor and this CD4, but also this CD4 glycoprotein, only then can this binding process take place. And once this binding process takes place between the B lymphocyte and our helper T cell, now we begin to initiate different types of defensive responses. What happens is the helper T cell begins to release lymphokines or lymphokines. And what these chemicals do is they basically initiate the process of cloning. So this B cell begins to clone itself, and when it clones itself, it produces identical cells, identical B cells that contain the same exact B cell receptor. And what that means is these cloned cells can then go on and find the same exact antigens floating around in our body. Now, what it also does is it induces the process of differentiation. Some of the cloned B cells begin to differentiate into plasma cells as well as into memory B cells, the specialized versions of our B lymphocytes. Now, what the plasma cells do is they begin to produce plasma soluble and mobile versions of these B cell receptors that can bind onto antigens. And what these are is our antibodies that we spoke of earlier. So antibodies are soluble and mobile versions of these B cell receptors that are found on the membrane of the B lymphocyte. And so what these plasma cells do is they contain highly extensive endoplasmic reticulum that can basically produce many of these antibodies. And then these antibodies can move within our blood system, our lymph system, and our tissue and bind onto that antigen and elicit some type of response, label that antigen for destruction by our immune system. Now, finally, these chemicals can also induce the differentiation of B lymphocytes into memory B cells. And memory B cells, instead of producing our antibodies, they store a copy of the antibody in case reinfection by that same pathogenic antigen ever takes place. So if the body is ever reinfected by that same pathogen, it's the memory B cells that will have a copy of that antibody and which will be able to recognize those cells immediately and essentially label them for destruction by our immune system. Previously, we focused on B lymphocytes, which are the cells that make up our humoral immunity. Now let's focus on T lymphocytes or T cells, which are the white blood cells that make up our cell. Previously, we focused on B lymphocytes, which are the B lymphocytes, or simply B. We produce many of these antibodies, and then these antibodies can move within our blood system, our lymph system, and our tissue and bind onto that antigen and elicit some type of response, label that antigen for destruction by our immune system. Now, finally, these chemicals can also induce the differentiation of B lymphocytes into memory B cells. And previously, we focused on B lymphocytes, which are the cells that make up our humoral immunity. Now let's focus on T lymphocytes or T cells, which are the white blood cells that make up our cell-mediated immunity of our adaptive immune system. Now, just like B cells, T cells also have special receptors on the membrane we call T cell receptors. And because there are two different types of T cell receptors, we categorize T lymphocytes into two categories. We have alpha beta T cells, which consist of special T cell receptors that have an alpha and a beta polypeptide subunit. And we also have gamma delta T cells, which contain T cell receptors that consist of a gamma and a delta polypeptide subunit. Now, it turns out that we know much more information about alpha beta T cells than we do about gamma delta T cells. And that's exactly why in this lecture, we're going to focus primarily on alpha beta T cells. Now, before we take a look at these alpha beta T cells, let's recall what an antigen presenting cell is, or an APC. So an antigen presenting cell is a type of white blood cell whose function is to engulf our antigens and take those antigens and present those antigens on the membrane of those APCs. So basically, these antigen presenting cells contain these protein complexes on the membrane known as the major histocompatibility complex. And what these protein complexes do is they're capable of binding these antigens that the cells actually engulf. Now, what the role of the alpha beta T lymphocyte is, it's to actually bind to these antigen MHC complexes once the antigen presenting cell actually engulfs that antigen and places it onto that MHC in the first place. So now, on top of having that T cell receptor that consists of an alpha and a beta subunit, these T lymphocytes also contain glycoproteins that are necessary to actually bind to the antigen presenting cells. Now, we have two different types of glycoproteins. We have CD4 glycoproteins and we have CD8 glycoproteins. And the difference between these two different glycoproteins is their ability to bind to different types of major histocompatibility complexes. So let's begin with the CD4 alpha beta T cell. So let's suppose we have the CD4 T cell, and what that basically means is it contains a T cell receptor that contains an alpha unit shown in purple and the beta unit shown in red. And it also contains a special glycoprotein that extends from the cell membrane, and that is shown in blue. Now, let's suppose our APC, the antigen presenting cell, such as, let's say, a macrophage, engulfs that pathogenic antigen, takes that pathogenic antigen, and places it onto the MHC class II complex. So this is the MHC class II complex, and this green part is our antigen. And once the antigen binds onto the MHC class II complex, only then can the CD4 alpha beta T lymphocyte actually approach and bind onto this complex, forming an interaction between the APC, the antigen presenting cell, and our T lymphocyte. So it turns out that T cells that have CD4 glycoproteins can only bind onto the major histocompatibility complex class II. And as we'll see in just a moment, we also have a major histocompatibility complex, class I, which these glycoproteins cannot actually bind to. Now, the question is, what exactly takes place when this binding is initiated? So basically, once the binding process takes place, the T cells can stimulate our immune system in two different ways. They can either stimulate the humoral immunity, or they can stimulate the cell-mediated immunity. So let's suppose the CD4 T cell that binds onto the APC is a helper T cell. In such a case, when this binding process takes place, and let's suppose the APC is a B lymphocyte, in such a case, the helper T cell, which is our CD4 T cell, will stimulate that B lymphocyte, our APC, to actually begin to undergo mitosis and create more clone cells, identical cells that contain the same exact B cell receptor. And they can also stimulate those B lymphocytes to actually undergo the process of differentiation and differentiate into plasma cells and memory B cells. So plasma cells are those cells that produce antibodies, while memory B cells store a copy of that antibody in case reinfection ever takes place. Now, instead of the CD4 being a helper T cell, let's suppose we have some other type of T lymphocyte that binds to, let's say, a macrophage. In such a case, we can actually stimulate the cell-mediated immunity. So CD4 T cells can also bind to APCs such as macrophages and dendritic cells, and they release these chemicals known as lymphokines, and these lymphokines will stimulate our cell-mediated immunity, and that means it will call upon other macrophages, other white blood cells that will come to that infected area and begin our defensive mechanism response engulfing those pathogenic agents. So our CD4 T cells can either stimulate the humoral immunity, or they can stimulate the cell-mediated immunity. On the other hand, as we'll see in just a moment, the CD8 T cells can only stimulate the cell-mediated immunity. So what exactly is a CD8 T cell? Well, a CD8 T cell contains that same alpha beta subunit, that same T cell receptor, but it contains a different glycoprotein. It contains a CD8 glycoprotein. And what the CD8 glycoprotein does is it is able to bind to the major histocompatibility complex class I and not the class II one. Now, the class I MHC is the protein complex that is used by our white blood cells to differentiate between healthy cells and infected cells of our bodies, our cells that have been infected by some type of viral or parasitic agent. So it turns out that CD8 T cells are those cells that help destroy the infected cells of our body. So once again, these T cells contain the CD8 glycoprotein next to their alpha beta protein receptor. These cells can only bind to the MHC class I complex, and recall that this is the complex that is used by our white blood cells to differentiate between healthy cells and infected cells. Now, one example of a T cell that contains a CD8 glycoprotein is the cytotoxic T cell, also known as the killer T cell, that is part of the cell-mediated immunity. So cytotoxic T cells are example of CD8 plus T cells. The plus simply means these cells have this glycoprotein. Now, generally speaking, these cytotoxic T cells and all CD8 T cells are those cells that can differentiate infected cells from healthy cells and kill off those infected cells. So to see what we mean, let's consider the following example. Let's suppose that we have the common flu. And what that means is a special type of viral agent known as influenza infected our body. Now, what this virus does is it infects our healthy cells and it creates infected cells. So the infected cell begins to produce the viral agents that are needed by the virus to essentially reproduce. But what the infected cell also does is it takes a piece of that virus, some type of antigen, and places it onto the major histocompatibility complex class 1 as shown in the diagram. So this is the MHC class 1 of the infected cell, and this green portion is the antigen that came from the viral agent that infected our cell. Now, once the cell is infected and once the infected cell displays the antigen, when the CD8 T cell, for example, the cytotoxic T cell approaches this, the CD8 glycoprotein can bind onto this MHC class 1 complex, and this T cell receptor recognizes this antigen. And when the binding process takes place, what the CD8 T cell usually does is it begins to release these very powerful proteolytic proteins that begin to drill holes and digest our membrane of that infected cell. And what that ultimately does is it kills off that infected cell and destroys it before that infected cell can infect other healthy cells of our body. In our discussion on T lymphocytes, we mentioned that there are two different types of T lymphocytes, and we can categorize these two different types of T lymphocytes based on the type of membrane receptor that they actually have. So one type of T cell is the alpha beta T cell, and we call this an alpha beta T cell because it contains a T cell receptor that has an alpha subunit and a beta subunit. Now, in our discussion on alpha beta T cells, we said that alpha beta T cells can only bind to either the MHC class 1 or the MHC class 2 complex found on some antigen presenting cell of our immune system. So recall that antigen presenting cells are cells such as macrophages, dendritic cells, and B lymphocytes that engulf antigens and then present those antigens to the T lymphocytes by using their major histocompatibility protein complexes found on the membrane. And we have these two different classes of these major histocompatibility complexes. The class 1 are responsible for differentiating between infected cells and healthy cells, and class 2 is used for communicating between different types of white blood cells. So alpha beta T cells can only interact with these antigen presenting cells. Now, what about the other category of T cells, known as the gamma delta T cells? So the gamma delta T cells contain T cell receptors that are slightly different. Instead of having an alpha and a beta polypeptide subunit, we have a gamma and a delta polypeptide subunit as shown in the following diagram. So the purple is our gamma and the blue is our delta. Now, we have a variable portion on our T cell receptor, the protein, and this basically varies from one cell to a different cell, and that's because this is the portion of the T cell receptor that contains that specific sequence of amino acids that can bind to some particular antigen. So it contains the antigen binding site. The constant region of our T cell receptor on the gamma delta T cell basically does not change too much when we go from one cell to a different cell. And this is the portion of our T cell receptor that actually binds onto the cell membrane of the gamma delta T cell. So we know that alpha beta T cells only bind to those antigens found on either the class 1 and class 2 major histocompatibility complex on these antigen presenting cells. But what about the gamma delta T cells? What types of antigens can these gamma delta receptors actually bind to? Well, it turns out that the gamma delta T cells don't actually need the MHC class 1 or MHC class 2 complexes to actually bind to our antigens. In fact, these gamma delta T cells can bind to fully intact antigens that have not yet been degraded by other white blood cells. For example, the macrophages and the other antigen presenting cells. Recall that these antigen presenting cells degrade, they break down the antigens and only display a small portion of that antigen on that MHC. But these gamma delta T cells don't have to rely on that single portion of the antigen. They can actually fully bind to a fully intact antigen in the same way that free-floating antibodies bind onto fully intact antigens. So gamma delta T cells can bind to any antigen that is not bound to the MHC class 1 and MHC class 2 molecules found on other white blood cells. And what that implies is they do not depend on antigen presenting cells to actually carry out some type of immune defense response. So we see that gamma delta T cells bind to antigens that alpha beta T cells cannot actually bind to. And what that means is our gamma delta T cells do not contain the CD4 and CD8 glycoproteins that are needed to actually bind onto the major histocompatibility complex, class 1 or class 2. Now, let's move on to the location where we normally find our gamma delta T cells is. So in the same way that our alpha beta T cells are produced in the bone marrow and mature in the thymus, the gamma delta T cells are also produced in our bone marrow and differentiate in our thymus. But these gamma delta T cells actually end up in the tissue of our body that interfaces with the outside environment. And that implies that these gamma delta T cells are found in the epithelial cells, in the epithelial tissue of our body, for example, in our skin, in our lungs, in our intestines, as well as other regions of our body. Now, what about the function? Well, because we don't know too much about these gamma delta T cells, we don't actually know what the exact function of these cells are, but we have a pretty good idea. Well, since these T cells do not actually bind onto the MHC class 1 or the MHC class 2 regions of these antigen-presenting cells, such as macrophages, dendritic cells, and B lymphocytes, that implies that gamma delta T cells do not actually depend on these antigen-presenting cells to carry out their immune defensive response. So although their function and mechanism of action is still not very well understood, they are believed to be part of the first line of defense and seem to react faster at a quicker rate than our alpha beta T cells do. In fact, these gamma delta T cells are believed to be involved with immunosurveillance. And what that means is basically swimming around the epithelial tissue of our body and making sure that everything is working correctly and no pathogen is found inside those regions of the body. So we see that the antigens to which these gamma delta T cells actually bind to are not only found on the actual pathogen, for example, our bacterial cell, but they're also found on infected cells of our body. For example, if we have some type of epithelial cell that is infected, for example, this cell, what the infected cell does is it displays an antigen on the membrane of that cell. And our gamma delta T cell can use the gamma delta T cell receptor to bind onto that antigen. And once bound, it basically begins to release special chemicals we call cytokines. And what these cytokines are believed to do is they carry out the following several functions. First of all, they begin the process of repairing these cells. They continue the process of immunosurveillance, as we discussed previously. They seem to indirectly affect the cytolysis of local antigenic presenting cells. So why would we want to lyse, for example, a macrophage? Well, if a macrophage eats too many of these pathogens, it will essentially stop functioning correctly. And at that point, we essentially want to lyse that particular cell. Now, these cytokines and these gamma delta T cells are all lysis of local antigenic presenting cells. So why would we want to lyse, for example, a macrophage? Well, if a macrophage eats too many of these pathogens, it will essentially stop functioning correctly. And at that point, we essentially want to lyse that particular cell. Now, these cytokines and these gamma delta T cells are also believed to regulate the influx of white blood cells to that specific area. So if this epithelial cell is infected, we want to bring more white blood cells to this area. And that's exactly what the gamma delta T cell is believed to do. Once it binds onto that antigen portion of the membrane, it initiates a response that essentially calls upon more white blood cells to this area so that the white blood cells can actually carry out an effective defensive response. Now let's compare and contrast the primary and the secondary response of our immune system to some particular pathogen. So let's begin by defining and discussing what a primary response is. So the first time that our body is ever invaded by some particular pathogen that carries its own antigen, our body, our immune system responds in a
All right, hold on, please. Hold on. Oh, give me 5 minutes. All right, Andrew. Let's get back, buddy. How many? 11. Those, and we continue on for more. Let me just see real quick. Which ones I'm missing again? Let's go back. Right. Yeah. So 1, 2, 3, 4. Oh, that's it. Wait, 1, 2, 3, 4. Okay, so 5. 6, 7, 8. Oh, yeah, okay. That's kind of actually quite there.
And this response we call the primary response. So let's suppose our pathogen makes its way into our tissues and releases its pathogenic antigen. Now, at this particular moment in time, we have never actually seen, our immune system has never actually seen this particular antigen. And what that means is we're going to have no corresponding antibodies in our body, in our blood, that can actually bind to that particular antigen. So at least in the beginning, our concentration of antibody that is specific for that antigen will be 0. Now, right away when we're infected, the innate immune system kicks in, and we can have the process of inflammation take place that essentially prevents that infection from spreading to other parts of our body. But our adaptive immune system will actually take time to take into effect. And so we're going to have to wait a certain amount of time for our adaptive immune system to mobilize itself and to actually create the appropriate lymphocytes that are needed to create those antibodies. So our adaptive immune system will need to create the appropriate plasma cells that can produce the antibodies that are specific to that infecting antigen. Now, once we have all those plasma cells, once we have all those active lymphocytes, then our antibodies will begin to be produced, and those antibodies will be released into our blood. And so the concentration of the antibodies in our blood will begin to increase sharply. So if we plot the concentration of antibodies that are produced versus time, we basically get the following diagram. So the y-axis is the concentration of our antibodies, our immunoglobulins, and the x-axis is the time given to us in weeks. So at week 0, we essentially have that pathogen invading our body. Infection takes place. Now, it's the first time that we are ever infected by that particular pathogenic antigen, and that means, at least initially, during the latent period, we're going to have a 0 concentration of antibody in our blood because we're not going to have those antibodies that can bind specifically to that infecting antigen. However, over time, when the adaptive immune system actually is mobilized, it will have those plasma cells that will be able to produce those antibodies. And in that moment in time, we have the logarithmic phase take into effect. And what the logarithmic phase describes is these plasma cells producing and releasing the antibodies into our blood. And so we see a sharp increase in our concentration of immunoglobulins. But eventually, we have this leveling off process taking place, and then we have the decline phase. And the decline phase takes place because our body is actually winning. Our antibodies are binding to the antigens, and they are labeling them for destruction. And so our immune system is able to destroy the infecting pathogens along with their antigens. And so that's why eventually over time, we see a drop in our concentration of immunoglobulins until it drops to very low undetectable levels as shown in the following diagram. And so this process ultimately takes place about 4 weeks as shown in this particular case for this particular invading pathogen. Now, the primary immunoglobulin that is used for this particular primary response, and in general, the major type of, and so we're going to have to wait, is its pathogenic antigen. Now, at this particular moment in time, pathogen. So now let's compare and contrast the primary and the secondary response of our immune system to some particular pathogen. So let's begin by defining and discussing what a primary response is. So the first time that our body is ever invaded by some particular pathogen that carries its own antigen, our body, our immune system responds in a certain way. And this response we call the primary response. So let's suppose our pathogen makes its way into our tissues and releases its pathogenic antigen. Now, at this particular moment in time, we have never actually seen, our immune system has never actually seen this particular antigen. And what that means is we're going to have no corresponding antibodies in our body, in our blood, that can actually bind to that particular antigen. So at least in the beginning, our concentration of antibody that is specific for that antigen will be 0. Now, right away when we're infected, the innate immune system kicks in, and we can have the process of inflammation take place that essentially prevents that infection from spreading to other parts of our body. But our adaptive immune system will actually take time to take into effect. And so we're going to have to wait a certain amount of time for our adaptive immune system to mobilize itself and to actually create the appropriate lymphocytes that are needed to create those antibodies. So our adaptive immune system will need to create the appropriate plasma cells that can produce the antibodies that are specific to that infecting antigen. Now, once we have all those plasma cells, once we have all those active lymphocytes, then our antibodies will begin to be produced, and those antibodies will be released into our blood. And so the concentration of the antibodies in our blood will begin to increase sharply. So if we plot the concentration of antibodies that are produced versus time, we basically get the following diagram. So the y-axis is the concentration of our antibodies, our immunoglobulins, and the x-axis is the time given to us in weeks. So at week 0, we essentially have that pathogen invading our body. Infection takes place. Now, it's the first time that we are ever infected by that particular pathogenic antigen, and that means, at least initially, during the latent period, we're going to have a 0 concentration of antibody in our blood because we're not going to have those antibodies that can bind specifically to that infecting antigen. However, over time, when the adaptive immune system actually is mobilized, it will have those plasma cells that will be able to produce those antibodies. And in that moment in time, we have the logarithmic phase take into effect. And what the logarithmic phase describes is these plasma cells producing and releasing the antibodies into our blood. And so we see a sharp increase in our concentration of immunoglobulins. But eventually, we have this leveling off process taking place. And then we have the decline phase. And the decline phase takes place because our body is actually winning. Our antibodies are binding to the antigens, and they are labeling them for destruction. And so our immune system is able to destroy the infecting pathogens along with their antigens. And so that's why time, eventually over time, we see a drop in our concentration of immunoglobulins until it drops to very low undetectable levels, as shown in the following diagram. And so this process ultimately takes place about 4 weeks, as shown in this particular case for this particular invading pathogen. Now, the primary immunoglobulin that is used for this particular primary response, and in general, the major type of antibody that is used for all primary responses is immunoglobulin M. So remember, we have 5 different classes of antibodies. We have 5 different types of antibodies. And one of these antibodies is immunoglobulin M. And it's immunoglobulin M that is produced predominantly in the primary response. Now, immunoglobulin M forms a pentamer. And what that means is 5 individual antibodies orient in the following format to basically create this pentamer structure, and they are held together by disulfide bond. So this is our primary response. Now, what about a secondary response? So let's suppose as soon as the primary response is over, our pathogen reinfects our body. So the same type of pathogen with the same exact antigens reinfects our body. Now, the type of response that our immune system will essentially elicit is called not a primary, but a secondary response. It's secondary because it's the second time our pathogen makes its way into our body. Now, the response will be different. The question is why? Well, because during the primary response, not only was the adaptive immune system producing plasma cells, but it was also producing memory cells. And recall that memory cells are those white blood cells that actually store a copy of that antibody for that specific antigen. And the reason the memory B cells store that antibody is in case reinfection actually ever reoccurs, actually ever takes place again. So what happens when the body is reinfected by the same type of pathogen? In this case, the immune system will elicit a secondary response, and it will be different than the primary response because of the presence of these memory B cells and memory T cells. So let's redraw this diagram as shown. So we have first infection taking place, and then we have the end of our primary phase, our primary response. And right when our primary response is over, we have the second infection taking place. So we are reinfected by that same exact pathogen that contains that same exact antigen. Now, because we have those memory B cells in our blood, circulating our blood, our lymph, and our tissue, we have that specific antibody that can actually bind and destroy that antigen. And so what that means is the latent period will be much shorter because our adaptive immune system already consists of those memory B cells and memory T cells that contain that specific antibody. And so the latent period will be much shorter. On top of that, the concentration of the peak, the highest amount, the highest concentration of immunoglobulins that we produce will be
antigens make their way into our body. The latent period. Was much less here, right? It should be much less.
Much greater than in the primary case as a result of the presence of those memory cells. And also, notice what happens in this phase. In this case, we have a very sharp decline in the concentration, and it drops to a very low, undetectable level. But in this particular case, it doesn't drop to a low value. It remains relatively high. And that essentially ensures that all that pathogenic antigen is completely destroyed by the antibodies in our blood. Now, in the case of the primary response, the major immunoglobulin was immunoglobulin M. But in the case of our secondary response, the major immunoglobulin is immunoglobulin G. So once again, following the first infection, the immune system will produce memory cells that will carry that copy of the antibody that is specific to that particular antigen. And when the antigen reinfects our body the second time, we're already going to have that that antibody circulating inside our blood. And so that will create a much quicker, a much more rapid response with a shorter latent period because of those memory B cells. In addition, the amount of antibodies that is formed will be much greater, and actually we're going to need a much lower concentration of antigen to elicit a secondary response than to elicit a primary response. That's another difference between the primary and the secondary responses. Right. So let's conclude by comparing and contrasting these two different types of responses. So for the primary response, we have a relatively long latent period, but for the secondary response, we have a relatively short latent period. So that means our secondary response is much quicker to take into effect as a result of those antibodies already being present inside our blood. Now, for the case of the primary, we form a relatively low concentration, peak concentration of antibodies, but here we form a much larger peak concentration of antibodies. So peak simply means the highest value of these particular hills. Now, notice that during our decline phase, we drop to a very low undetectable value in the primary case, but in the secondary case, we drop to a higher value. And because we have a higher value of antibodies during our decline phase, the secondary immune response will be much more efficient in actually binding the antibodies onto our antigens and destroying those antigens. And finally, we see that in the primary response, we use immunoglobulin M, but in the secondary response, we use immunoglobulin G. Now, the final difference is to initiate a primary response, we need a relatively high amount of antigen in our body, but to elicit the secondary immune response, we actually need a much lower concentration of antigen inside our blood. Now let's focus on active immunity and passive immunity, and let's discuss a concept known as immunization. Now, let's begin by discussing how the natural process of infection takes place and what our body does to fight off that infection. So let's suppose we have a cut in our skin and the pathogen makes their way into our body. What happens is infection begins, and as soon as infection begins, the innate immunity of our immune system kicks in and it begins the process of inflammation. Now, what inflammation does is it ultimately aims to block off that infection, to localize that infection to a specific location in our body, and that prevents that infection, the pathogen, from actually spreading to other parts of our body. Now, at the same time that inflammation is taking place, the adaptive immunity of our immune system begins to mobilize, it begins to activate itself. And what happens is special white blood cells of the adaptive immunity begin to form. So we have plasma cells and we have memory cells. Plasma cells are those white blood cells that produce antibodies specific to that invading antigen. And these antibodies are released into our blood, our lymph, and our tissue. And when the antibodies bind onto that antigen, they label that antigen for destruction by other white blood cells. And so that calls upon macrophages, natural killer cells, cytotoxic T cells, and many other different types of white blood cells to that area, and that destroys that pathogen along with its antigen. Now, this is basically how our immune system defends and protects our body from these invading pathogens. Now, following our infection, our body develops something called active immunity. And what that means is our body develops these memory cells. And these memory cells contain a copy of the antibody that is specific to that antigen that infected our body in the first place. And what that is useful for if we are ever reinfected by that same pathogen that contains that same antigen, we have these memory B cells that can now initiate a very quick and a very rapid response, and we actually won't feel the same exact effect that we felt the first time when we were infected by that pathogen. So the process of active immunity is a process in which our body develops immunity, these memory cells, to the pathogen by being directly exposed to our antigens. For example, people who are exposed to chickenpox gain active immunity and will be very unlikely to ever feel that same effect they felt the first time when they were exposed. Now, there are two processes by which we can gain active immunity. What we just discussed a moment ago is a natural process. So active immunity can be achieved either naturally through some type of exposure to that pathogen, for example, an infection, which is what we discussed earlier, or we can actually induce active immunity artificially via process known as immunization. And immunization involves injecting something called vaccines into our body. So what exactly is a vaccine? So a vaccine is essentially this little test tube that contains some sort of pathogen inside that test tube. Now, the pathogen has been altered in some way or form. It has been inoculated. And what that means is we change that pathogen in the laboratory, and we change it in such a way to ensure that it does not damage the body as severely as it would normally when we are infected by that pathogen in a natural way. Now, in both of these cases, in the natural case and in the artificial case, we obtain the same exact result. We essentially either inject or somehow obtain these antigens. Our immune system begins to develop antibodies as well as memory cells that contain a copy of that antibody in case we are ever reinfected. And that's how we develop something called active immunity. So vaccines are normally developed in laboratories by many different methods that we're not going to focus on in this lecture. Typically, the pathogen is inoculated, which means that it is changed in some way or form as to ensure that not too much damage is actually taken by the body so that we don't feel the same exact bad effect that we would normally feel in the natural process of infection. And in many cases, receiving a vaccine essentially creates no effects whatsoever, no effects that we can actually visibly see or physically feel. Now, to further prevent us from actually experiencing any form of discomfort during vaccination, what medical researchers do is they essentially kill off that pathogen altogether, and they only take a very small portion of an antigen of that pathogen. So they essentially cut that pathogen and use only the epitope, the antigenic determinant of that antigen, because it's that part of the antigen that ultimately binds to the antibodies of our body. And in this way, the body can still produce antibodies following vaccination. And at the same time, it will create a very, very little effects that will actually hurt the body. Now, everything we discussed so far is active immunity, and active immunity involves developing our memory cells. And we said we can gain active immunity either artificially or either via natural means when infection actually takes place. Now let's discuss something called passive immunity. Passive immunity basically means we do not develop any memory B cells or memory T cells. And that's because we actually inject antibodies directly into our blood, into our lymph, or into our tissue. And by injecting the antibodies, we're not exposing the immune system to our antigens. And so we're not giving our immune system, the adaptive immunity of our immune system, the chance to actually develop those antibodies themselves. So another form of immunity is called passive immunity. In passive immunity, antibodies are developed in the laboratory that are specific to some particular pathogen. And these antibodies, when they're injected into our body, they begin to circulate in our blood system, in our lymph system.
Antibodies transfer via placenta or in breast milk. Infection of pathogen. Injection of antibodies.
And in our tissue. And if we are exposed,
Vaccination. Ooh, artificial, active, artificial, passive.
to that pathogen that carries that antigen that is complementary to the antibody that was injected into our body in the first place, those antibodies will bind onto those antigens. The artificial antibodies will bind onto the antigens, and that will initiate a response in which it will label those antigens as well as the pathogen for destruction by our white blood cells. Now, one very important point about passive immunity, once injected into our body in the first place, in our blood system, in our lymph system, and in our tissue. And if we are exposed to that pathogen that carries that antigen that is complementary to the antibody that was injected into our body in the first place, those antibodies will bind onto those antigens. The artificial antibodies will bind onto the antigens, and that will initiate a response in which it will label those antigens as well as the pathogen for destruction by our white blood cells. Now, one very important point about passive immunity, once again, is it does not develop memory cells. And so this means that eventually these antibodies will be degraded inside our body and they will cease to exist. And at that point, we will lose our immunity. And that's exactly why this is called passive immunity. Now, one very important point about passive immunity, once again, is it does not develop memory cells. And so this means that eventually these antibodies will be degraded inside our body and they will cease to exist. And at that point, we will lose our immunity. And that's exactly why this is called passive immunity. It only persists for several months following injection of those antibodies, while active immunity usually persists for the duration of our lifetime. So we saw that active immunity can be acquired naturally or artificially. And so far we discussed the artificial process by which we gain passive immunity. So we actually synthesize those antibodies in a lab. Now, it turns out that we also have a natural way to obtain passive immunity, and this takes place in a woman when a woman is pregnant with a child. So passive immunity can also be developed naturally. Pregnant women give their developing fetus passive immunity by producing specific antibodies known as immunoglobulins G. And immunoglobulins G are small enough to actually pass across the placental membrane and into the blood found inside our fetus. And what that gives the fetus is passive immunity to any type of antigen or pathogen that might infect that fetus. And this is important because as the fetus is developing, his or her immune system is also developing. So it's important that during this process of development, our fetus has an ability to fight off an infection that infects that mother. Now, another way we pass down or we create passive immunity naturally is via the process of breastfeeding. So the milk that is found in women during the process of breastfeeding contains yet another type of immunoglobulin antibody known as immunoglobulin A. And once our child gains the immunoglobulin A via the milk, via the process of breastfeeding, that boosts their immune system because the child's immune system is still developing following childbirth. So this is basically a diagram, a flow chart that describes everything we just discussed. It summarizes our results, our discussion. So we have our immune system that is split into two categories. So the innate immune system that begins to act immediately after infection. It involves cells like macrophages, neutrophils, and so forth. And then we have our adaptive immunity that develops over some period of time because it has to synthesize the plasma cells, the memory cells, as well as other cells such as helper T cells and cytotoxic T cells. Now, adaptive immunity can be split into two. We have artificial and we have natural. And each one of these can be split into active and passive. So basically, during the process of vaccination, during the process of immunization, when we take a vaccine and we inject it into that organism, into that individual, we inject an inoculated form of that pathogen. And what that does is it gives us active immunity by not having to experience the same exact detrimental effects that we would naturally experience if we are infected in a natural way. Now, in this process, we gain memory cells. If we examine the natural process, we have to get the actual infection from that pathogen. We have to be infected by that pathogen to gain the same exact response to basically form those same memory cells that contain the antibody. So both of these two are two methods of gaining active immunity. One is via an artificial, so immunization via a vaccine, and the other one is a natural via the process of infection. Now, what about the temporary process of passive immunity, which basically injects in one way or form antibodies into our body, and those antibodies can bind onto the pathogenic antigens, labeling them for destruction. So we also have artificial and natural. Now, artificial includes actually taking those antibodies, injecting those antibodies into our body, and allowing those antibodies to circulate our system. Now, the problem with this is it doesn't produce any memory cells. And so after several months, we essentially lose any immunity that we initially had as a result of that passive immunity. And we also have the natural process of passive immunity, as we discussed just a moment ago. So antibodies transfer via the placenta when the woman is essentially pregnant, or during the process of breastfeeding when the milk is transferred to that child, along with those immunoglobulins, specifically immunoglobulin A. So we conclude that immunization is a very useful process of actually giving our body active immunity, the ability to fight off infections if we are ever actually infected with some particular viral or bacterial agent. For example, we have vaccinations for things like polio, botulism, tetanus. We also have vaccination for the whooping cough, as well as many, many other lethal pathogenic infections and pathogenic agents. Nowadays, many different types of tissues and many different types of organs have been successfully transferred from one individual to a different individual, and this process is known as grafting or organ transplant. Now, many different examples of successful organ transplants exist. For example, we've been able to transplant organs such as our heart, our lungs, our liver, our kidneys, our bone marrow, the ovaries, as well as things like our blood, which we'll focus on in the next lecture, and the cornea of the eye. And actually, the cornea of the eye is a relatively successful type of grafting process because the cornea represents an immunologically privileged site. And that basically decreases the complications that are involved with grafting of the cornea. In fact, we can successfully remove the cornea from a cadaver and place it onto a living individual. Now, when we transfer an organ or a tissue from one individual to a different individual, in this case, that organ or tissue is known as an allograft. But if we're removing a tissue from one individual and moving it onto a different location on that same individual, allograft. But to a different individual, in this case, that organ or tissue is known as an allograft. But if we're removing a tissue from one individual and moving it onto a different location on that same individual, in this case, we call that organ or tissue that is being removed and transferred an autograft. So auto means we're dealing with that same individual, and allo means we're dealing with two different individuals. Now, as you might expect, the process of grafting and organ transplantation is actually a very, very complicated process, and that's because it deals with a great amount of precision, a great amount of analyzation, and a great amount of preparation. Now, the primary thing that creates these complications is actually the immune system of that host individual that is accepting that graft or that organ. So let's discuss some of the major issues involved with transferring grafts as well as organs from one individual to a different individual. So let's examine graft rejection, let's discuss graft versus host disease, and also infections. And then let's discuss some of the methods that medical professionals actually use to fight these different complications that arise from transplantation. So let's begin with graft rejection. And let's first recall how our immune system actually works. So recall that the entire goal of our immune system, the entire goal of white blood cells, is to be able to differentiate between our own host healthy cells and infected cells or pathogens that might make their way into our body. So our white blood cells use something called the major histocompatibility complex and self-antigens to basically distinguish our host cells from infected cells or pathogens. Now, the problem with grafting or transplantation is when we actually transfer that tissue or that organ, the tissue or the organ on the other individual might have different major histocompatibility complexes and different self-antigens. And if these antigens are not compatible, if there is no match, then what the white blood cells of that host immune system do is they recognize those other self-antigens of the other individual as pathogenic, as being foreign, and they mount a defensive response. They label those cells for destruction and begin destroying that allograft altogether. So once again, when a tissue or organ is transplanted, it has a very high chance of being rejected by the host individual. Recall that our immune system attacks anything that it recognizes as foreign or pathogenic. And if the major histocompatibility complex, MHC, self-antigens of the transplanted tissue cells do not match or are not compatible with the host cells, then the host immune system will mount a defensive response and destroy that allograft. And we see that take place in the following diagram. So let's suppose this is our host cell, and the host cell will contain special major histocompatibility complexes on the membrane as shown in blue. And it will take some type of protein, some type of self-antigen, and place it onto that complex as shown in green. And the host immune cells, for example, cytotoxic T cells, will be able to distinguish this host cell from...
How is it? I forgot. How does it know it's
Yeah, it's a self-antigen presenting. Yeah, antigen presenting. Presenting. Okay. Yeah.
Some type of foreign pathogen as a result of these self antigens present on the MHC protein. Now, if we basically transplant a graft, a tissue, or an organ that contains cells with a different MHC protein with a different major histocompatibility complex that contains an incompatible, a different self antigen, then the host immune cell will label this cell as being pathogenic, as being foreign. And what will happen is this cell will destroy that graft cell along with that entire allograft. And what this cytotoxic T cell does specifically is it binds onto this complex and it initiates the release of powerful digestive proteins. And those proteins drill holes in the membrane of these graft cells, and that ultimately lyses and destroys the cell. So this is the major problem that is involved with the transplantation and grafting process, the actual immune system of that host organism, host individual, destroying that transplanted tissue or organ. Now, another important complication is actually the opposite of this. So this is called graft versus host disease, or GVHG. So let's suppose we're transplanting some type of tissue or organ from one individual to a different individual, and this particular organ contains a high concentration of T cells, of white blood cells. And one example of such an organ, such a tissue, is bone marrow. So let's suppose we're transferring bone marrow from one individual to a different individual. The problem with this is the bone marrow contains a high concentration of T cells. And these T cells, when they're transferred into the host individual, the T cells contain receptors that might not recognize the self antigens and the major histocompatibility complexes found on the host cells. And what that means is when the graft cell binds onto the host cell, it will begin to release these enzymes that will destroy the host cells of that host individual. So when transplanting tissue that contains a high concentration of white blood cells, such as T cells, there's a high probability that the graft T cells will recognize the host cells of that individual as being foreign, as being pathogenic. And this will begin a defense response in which these T cells will begin attacking and destroying our host cells. And this is of particular importance when transplanting things like the bone marrow, because bone marrow actually contains a very high concentration of T cells. So these are the two major problems, two major complications that accompany grafting and organ transplant. Now, another complication that is really no longer a complication because we have ways of dealing with it is infections. But this was a problem when organ transplant and grafting was at its beginning. So the donated tissue or organ may contain dangerous pathogens such as HIV, hepatitis B or hepatitis C, rabies, syphilis, and many other different types of diseases and pathogens. So basically, if we're transplanting an organ that contains HIV into an individual that does not contain HIV, that individual will obtain that HIV and will then be HIV positive. Now, this was a problem in the beginning, but nowadays we have different types of ways of testing for these different types of viruses and pathogenic agents. And so before the donation or transplantation actually takes place, we know what types of pathogenic agents are found in that donated tissue or donated organ. Now, the next question is, how exactly do we solve these different complications? What exactly do we do? Well, the best case scenario, the best way to actually ensure that the organ or tissue that is transplanted is successfully accepted by that host individual is to actually use the same tissue of that individual. Now, of course, this cannot be done always. We can, for example, easily transplant a piece of skin from one location to a different locations in our body, but obviously we can't transplant things like the heart because we only have a single heart. Another good case scenario is if we have a twin. If we actually have an identical twin, this creates very little complications because those two twins will have very similar, if not identical, MHC, major histocompatibility self-antigen complexes. And so when we transplant an organ or a tissue from one twin, from one of the twins to different twins, this will create very little complication and the immune system will accept that organ. Now, of course, these instances are actually very rare because not always do we have a twin and not always can we actually transplant an organ because we only have one of each organ, not including things like our lungs or our kidneys. Now, two very common methods, much more common methods than what we just discussed, are tissue typing and immunosuppression. Now, tissue typing is actually a pretty complicated process, so we're not going to go into too much details, but what tissue typing basically is, it's a process of determining the type of major histocompatibility complexes and the type of self-antigens that are found within that host individual, and then finding the exact match or a very similar match, a compatible match from another individual that contains very similar MHC antigen complexes. So tissue typing is the process that involves determining the major histocompatibility complex antigens of the host individual and finding a donor that is most compatible, that contains the least mismatches. Another important type of method that is used to basically solve these complications and is probably used virtually on all cases of grafting and organ transplant is immunosuppression. Now, immunosuppression deals with using different types of chemical agents that are extracted from other organisms or synthesized in a lab. And what these chemical agents do is they essentially decrease the ability of our immune system to actually fight off different infections, including these grafts and organs that are transplanted into our body. So these chemical agents interfere with the processes of forming white blood cells. For example, they can interfere with the process of protein synthesis or DNA replication, and that will greatly hinder the ability of our immune system to actually form any type of white blood cell. And so that suppresses the immune system and prevents it from actually mounting a defensive attack against that transplanted tissue or organ. Now, the major problem with this immunosuppression process is it's actually very dangerous. So as soon as we suppress our immune system, we decrease its ability to fight any type of infection, and that includes a simple infection from the common flu or the common cold. So because our immune system is suppressed, it not only is not capable of fighting that graft, but it also can't actually fight off any type of infection. So people that are immunosuppressed can sometimes die from the common cold or the flu because their immune system is not capable of mounting any type of defensive response. On top of that, immunosuppressed people are also much more likely to actually develop cancer. And that's because cells like natural T cells and cytotoxic T cells are the cells that fight off the cancer developing cells. And because our system is immunosuppressed, that means those cells cannot suppressed people are also much more likely to actually develop cancer. And that's because cells like natural T cells and cytotoxic T cells are the cells that fight off the cancer developing cells. And because our system is immunosuppressed, that means those cells cannot find those cancer cells, and therefore the cancer cells that come from healthy cells can develop, and that can progressively lead to some type of cancer. So although this method is actually helpful in fighting off these complications and solving these complications, this method is also very dangerous in itself because it depresses our immune system, and that can basically lead to an infection or cancer that can ultimately kill that individual. Now that we discussed how our immune system actually works, let's discuss the types of blood groups or blood types found in humans. And then let's briefly discuss the process of blood transfusion. Now, humans contain 4 different types of blood groups. So we have blood type A, blood type B, blood type AB, or blood type O. Now, the first question is, what exactly determines the difference between these different blood types? And more specifically, what determines the blood type of any given individual? Because any given individual can only have one of these 4 different types of blood groups. Now, it turns out that on chromosome 9 of our karyotype, we basically have the allele, the gene that codes for a special protein membrane. That chromosome 9 of our karyotype, we basically have the allele, the gene that codes for a special protein membrane that is found on red blood cells. So each human inside our DNA, we have a gene that codes for this protein. Now, before the protein is actually attached onto the membrane of red blood cells, it is modified in the Golgi apparatus, and a carbohydrate attachment is made onto that protein to form a glycoprotein. And this glycoprotein then is transferred onto the membrane of red blood cells. And it's the terminal sugar on that glycoprotein that determines the type of blood group or blood type that our individual actually has. So basically, let's take a look at the following diagram to see exactly what we mean. So we have the phospholipid bilayer of the red blood cell. This is our cytoplasm portion, and this is the extracellular matrix. So this is the protein component of the glycoprotein, and these purple extensions are the sugar components. And it's the terminal sugar of that glycoprotein that determines the type of blood type or blood group that our individual has. Now, these glycoproteins are also sometimes known as self-antigens or simply antigens, as we'll see in just a moment. And we'll see why that's the case. It has to do with our immune system. Now, there are two different types of glycoproteins. So there are two different types of antigens. One of them we call antigen A or glycoprotein A, and the other one we call antigen B or glycoprotein B. And it's the presence or absence of these glycoproteins that determines and differentiates these blood groups from one another. For example, blood type A means our red blood cells contain antigen A and not antigen B. Blood type B means our red blood cell membrane contains antigen B but not antigen A. Blood type AB means our red blood cells contain both antigen A and antigen B. And finally, blood type O means we have neither antigen A or antigen B on the red blood cell of the membrane of our red blood cell. Now, the next question is, what exactly is the big deal? And what's the relationship between these groups and our immune system? Well, let's begin with blood type A. So in an individual that has blood type A, what that means is on chromosome 9 of that individual, they have a gene that codes for antigen A. And that means all the red blood cells will contain this particular glycoprotein, antigen A. Now, the immune system, because these are self-antigens, that means the immune system will not produce any antibodies against the antigen A glycoprotein. But because the red blood cells of blood type A individual do not have antigen B proteins, what that means, our immune system will begin producing antibodies against that particular antigen B. And so if that individual with blood type A ever receives blood type B, for example, the red blood cells of the blood type B individual will be destroyed as a result of the presence of those antibodies against antigen B. And we'll see exactly what that means in just a moment. So this is our red blood cell of a person that has blood type A. So blood type A individual contains red blood cells that contain a membrane protein we call antigen A. And so that individual will not produce antigen A antibodies. They will only produce antigen B antibodies because they do not have any antigen B proteins on the membrane. On the other hand, let's focus on the blood type B. So if an individual has blood type B, that means they have a gene that codes for antigen B, and all the red blood cells will contain membrane-bound antigen B glycoproteins as shown in green. And so this will be the self-antigen, and what that means is our immune system will not produce any antibodies against this antigen B, but now will produce antibodies against antigen A because it doesn't have any antigen A on that protein. So this is blood type A, and this is blood type B. Blood type A contains antigen A and antibodies against antigen B, while blood type B contains our membrane-bound antigen B and so contains antibodies against antigen A. Now, the next question is, how exactly do we pass down these blood groups from one individual to our offspring? So basically, each parent has the ability to donate a gene for that particular blood group that they actually have. So let's suppose this is the male parent chromosome, this is the female parent chromosome.
Oh, ideally, yeah. Receiving. Oh, actually, that's not true. No, actually, the best thing to be is blood type AB.
So
not blood type O, I'm sorry. Blood type AB is the best thing to be. Yeah.
And this is chromosome 9 for each case because this is the allele, the gene section that codes for a particular type of blood group. So this is the blood type allele. Now, as we mentioned earlier, the ABO blood group locus is located on the chromosome number 9, and each allele is passed down from each one of the two parents. Now, as it turns out, the blood type is a codominant trait, a codominant trait. And what that basically means is if one parent gives an antigen A gene while the other parent gives an antigen B gene, they will both be expressed on that red blood cell's membrane. And what that means is if this is A and this is B. When they combine, they will form the AB blood type group. And what that means is the red blood cells will have both of these antigens attached onto the membrane of the red blood cell. And this is what we call blood type.
Interesting. So if you're, okay, that's pretty cool to know. It's a codominant what, trait, allele, codominant allele.
AB. And because in this individual, the red blood cell membrane contains both of these antigen types, our immune system will not produce antibodies against both of these types. And that's because the immune system will see antigen A and antigen B as self-antigen. So these individuals with blood type AB do not have antibodies for either antigen A.
Right. Yeah, alleles. Yes.
Or antigen B. Now, it is also possible that our individual doesn't have this gene on chromosome 9, and that means if it doesn't have that gene, it cannot actually express and produce any one of these antigens. And in such a case, if the male parent doesn't have that gene and the female parent doesn't have that gene, and they essentially mate, they will produce an offspring, an individual in which we basically have neither antigen A nor antigen B on the membrane. Now, it is also possible that our individual doesn't have this gene on chromosome 9, and that means if it doesn't have that gene, it cannot actually express and produce any one of these antigens. And in such a case, if the male parent doesn't have that gene and the female parent doesn't have that gene, and they essentially mate, they will produce an offspring, an individual in which we basically have neither antigen A nor antigen B on the membrane of that red blood cell. And in that case, our immune system will produce both antibodies against antigen A and against antigen B, and this is what we call a blood type O. So such an individual lacks both antigens and their red blood cells on their red blood cells, and so the immune system produces antibodies against antigen A as well as against antigen B. So now that we discussed the 4 different types of blood types, blood groups, let's discuss the process of blood transfusion. So it is actually possible to transfer blood from one individual to a different individual. However, one must keep in mind that certain blood types cannot be mixed. And that's because if they are mixed, they will agglutinate. And what that means is antibodies will combine with their complementary antigens, and they will basically clump together, and the blood will be rejected during that transfusion process. So this is because people carry antibodies for antigens they don't have. And so mixing red blood cells that have particular antigens with complementary antibodies will cause them to stick together, causing the process of agglutination. So to see exactly what we mean, let's take a look at the following table, which basically describes which blood types we can mix and which blood types we cannot mix. So on this table, this row, the red row, basically describes the blood of that individual that we are donating, while this column, the blue column, basically describes the blood of that individual that is receiving that donating blood. So this is our receiver, it's the recipient, and this is the person that is donating that blood. Now, yes means that by mixing these 2 blood types, we basically do not have agglutination taking place, and so mixing is allowed. But no means that we cannot mix these two blood types because agglutination will take place. An antibody will bind to an antigen, clumping will take place, and so our blood will be rejected. So let's begin with A. Let's suppose our individual that is receiving that blood has blood type A. And what that means is it contains antigen A on the red blood cells and it produces antibodies against antigen B. And so what that means is if the donating person has the same blood type A, then that means that person also has these red blood cells with the same type of antigen A. And so when we mix these two bloods, they will readily mix and no agglutination actually takes place. Now, what about if we donate blood from blood type B? Well, blood type B contains antigen B and forms antibodies against antigen A. And by mixing our B and A, we basically have the process of agglutination taking place. The antibodies against antigen B in the receiver will attack the red blood cells that basically come from this donating person that has blood type B. Now, what about AB? Well, AB is the same exact case. On AB red blood cells, we have both antigen A and antigen B. And since this produces antigen B, antibody B, the antibody B will bind onto AB red blood cells, destroying those red blood cells, forming this agglutination process. And so this and this is not allowed. But if we take an O individual and we donate blood from the O individual to an A, because the O individual doesn't have either antigen A or antigen B, what that means is the antibody against antigen B that this individual blood type A has will not be able to bind to the red blood cells of type O person. And so this process of mixing will take place. Now, notice one important point about blood type O. If we examine this entire column, we have yes all the way down. And what that means is this is the universal donor. A person with blood type O can donate to any individual, and that's because their red blood cells have neither antigen A nor antigen B, and any antibody against A or B will not be able to bind to these red blood cells. And so we can easily donate type O blood to either one of these four red blood types. On the other hand, this O cannot actually receive any blood unless it's type O. And that's because if, let's say, our person has type O red blood cells, that means they produce both antibodies against antigen A and antigen B. And by donating A, B, or AB to this person with blood type O, that means the blood type O person will have antibodies that will kill off all these different red blood cells, A, B, and AB. And only this one, the blood type O donator, will not have the red blood cells with either of these antigens, and so the antibodies will not affect that person. So you can examine this table in more detail to basically convince yourself that this transfusion system actually works. Now, the final thing I'd like to briefly talk about is the type of genotypes that form the different types of blood types. So let's take a look at this table here. So this column describes our blood types, so A, B, and O. And this describes the potential genotype that we must have to basically form these types of blood types. So let's begin with blood type A. So if our male parent has the A blood, and if the female has the A blood, they will mix, they will produce blood type A. Now, if the male has, let's say, the blood type A, and the female lacks either one of those traits, either one of those codes, what that means is we'll have IA and lowercase io. And what that means is we'll still form blood type A because the offspring will have the gene from the male parent that will create that protein, antigen A. Now, the same thing is true for blood type B. IB and IB forms blood type B, and so does IB, lowercase io. Now, what about blood type AB? So to form blood type AB, the male parent has to be A, the female parent has to be B, or vice versa. This male has to be B, and this female has to be A, and only then will we form blood type AB. And finally, to form blood type O, both of these have to be recessive, meaning they both have to lack these genes that code for either one of these antigens. And so in that case, if we have recessive and recessive, we form lowercase io, and that gives us blood type O. So these are the four different types of blood groups that humans actually have. And we see that knowing how the immune system actually works and the fact that it produces antibodies is crucial to actually understanding how and why blood transfusion actually takes place and how it works. Aside from antigen A and antigen B, which can both be found on the membrane of red blood cells and which actually determine the type of blood group that the individual has, another group of antigens that can also be found on the membrane of red blood cells is a group we call the Rh factor. Now, by far the most common type of antigen within the Rh factor group of the human body is known as antigen D. In fact, about 85% of the individuals living in the United States that came from Western Europe have a gene on their DNA that codes for the antigen D protein. So, an individual with the gene on their DNA that codes for antigen.
I'll be back in the restroom. Hello? Oh. Hi, Nadine. Nadine? Greetings from Algeria. What are you studying? I'm studying immunology. Oh, immunology. Yeah. Studying immunology. And I'm almost done. Studying immunology, we're almost done. Then I have then we'll study. I don't know if we have to go over it again. I don't know. Maybe not. Let me see. Depends. It's because it's school. Yeah, actually, I think I will. Yeah, I think I still will. Yeah, then we'll study biochem, hemoglobin. Hemoglobin and hemoglobin. And embryology. Embryology. We'll do one quick video. Association curve. And I want to do prokaryotes. Viruses and bacteria. Yep. And then I have a whole list of other things that that I need to study. Actually, maybe I should just write them down now, huh? So I don't forget for later. I can write it in the back of this one, I guess. Where's my black pen? Oh, it's right here. Overview metabolism. waves. Wait, that's another one? Yeah. Cycle. Process. Inspiration, fat and protein. Fat and protein metabolism. Fat and protein metabolism. And excuse me. Gastrointestinal system. Regulation of metabolism. Renal system. Renal regulation of blood pressure. Reproductive system. Skin system. Skeletal system. Oh my goodness. That's the skin, right? Yeah. It's got more to go. I guess not. Interesting. Maybe it's. I'm not sure. Oh, I think it's touched upon in. I think it's most likely touched upon in psychology, then, huh? Possibly. Yeah. These are like really, really quite short. I don't think I have a problem with this. It pretty much ends it on psychology. Okay, anyways. Let's go back to where we were. Well, where we'll be is overview of metabolism when I start. Which is somewhere in the beginning. Here. Yeah. I'll just actually put it on. Right. I think so. I don't think I missed anything. Yeah, no, I don't think we did. I don't think we did. Yeah. Okay. Sure. Sure. And I'll be back. Be right back. MEP. from oh, thank God. Oh, oh no. I think so. It's being done.
D is known as an Rh positive individual. And what this means is an Rh positive individual will have the gene to create the antigen D protein. So their red blood cells will contain that antigen D. On the other hand, an individual that is said to be Rh negative does not have the gene on their DNA and so will not code and create that protein and will not have antigen D on the membrane of their red blood cells. So in this particular diagram, we're describing a person that is blood type A and which is Rh negative. Now, unlike the ABO blood group, an Rh negative individual will not normally produce the antibody against antigen D. However, if they are actually ever exposed to that antigen D, only then will their immune system begin producing the antibody against antigen D. And to see what we mean, let's once again look at the following diagram. Let's suppose we have an Rh negative individual that has the blood group A, blood type A. What that means is they will have antigen A on their membrane and will not have antigen B. And it also means that because they don't have antigen B, their immune system will readily produce antibodies against antigen B, and those antibodies will circulate in the blood. Now, what it means for the individual to be Rh negative is the following. They don't have that antigen D on their membrane, but their immune system will not necessarily produce the antibody against antigen D. The immune system will only begin producing that antibody against antigen D if they are actually exposed to that antigen D. And we'll see an example of this in just a moment when we discuss the process of pregnancy and childbirth. So now, it turns out that the gene that codes for the Rh factor, the protein antigen D, is actually dominant to the recessive trait, to the gene that does not code for it. So to see what we mean by that, let's suppose we have a father that is heterozygous for that trait. So we have a dominant uppercase R and a recessive lowercase r. And let's suppose the father mates with a mother, a female, that is also heterozygous for that same trait. So uppercase R, lowercase r. So we have the father and the mother, and they have an offspring, a child. Now, what this describes is the probability of the child being Rh positive or Rh negative. So because the gene is dominant to the recessive trait, that means uppercase R and uppercase R will be Rh positive, and so will uppercase R and lowercase r, because uppercase R is dominant over lowercase r. So we have 1, 2, 3 out of 4 will be our Rh positive. And so 75% will be Rh positive, and there's a 25% chance that it will be Rh negative. So we get Rh negative only if we have lowercase r, lowercase r. Now, instead of mating a father who is uppercase R, lowercase r, and a mother who is uppercase R and lowercase r, let's suppose a woman who is Rh negative, so lowercase r, decides to have a child with a male who is Rh positive. Now, we know there are two different types of Rh positives. We have uppercase R, which is homozygous dominant, and we have the heterozygous as shown. So let's suppose we have a man who is heterozygous and a female who is homozygous recessive. And we have a female who is homozygous recessive and a male who is homozygous dominant. In both cases, when these individuals mate to produce a child and offspring, there is a probability that the offspring will be Rh positive. In this case, there's a 50% chance of the child being Rh positive. So we have 50%, and 50% of the child being Rh negative. But in this case, it's 100% likelihood that the child will be Rh positive. So let's assume for the time being that the fetus that is produced is actually Rh positive. And that means that inside the fetus, inside the blood of the fetus will have red blood cells that contain the antigen D on the membrane of those red blood cells. The question is, how will this actually affect that pregnancy, and how will it affect any future pregnancy that the woman may have? So to answer this question, let's begin with the following diagram. So this is our fetus. This is the placenta that connects the bloodstream of the fetus to the bloodstream of our female, of the mother, and this is the blood vessel of our mother. Now, normally, because the red blood cells are too large, they cannot pass across the placental membrane. But during pregnancy, during childbirth, that is, there is a small likelihood that there can be exchange of red blood cells. There's a leakage of red blood cells, and some of the red blood cells can be exchanged between the fetus and our woman. Now, we know that because the woman is Rh negative, that means within the bloodstream, the woman will not have any antibodies against antigen D because that woman was never exposed to antigen D before that, and so she is Rh negative. But because the child, the fetus, is Rh positive inside their blood, they do have red blood cells that contain antigen D. And when during childbirth, some of these red blood cells of the fetus leak into the blood system of the mother, some of these antigen D red blood cells end up in the mother's bloodstream. And now, because the mother is exposed to antigen D for the first time, the immune system will kick in and begin producing antibodies against the antigen D found on those red blood cells that essentially leak into the mother from the fetus. Now, this will not actually affect this fetus in any way because this process takes place during and after childbirth, but it will affect any further pregnancies that the mother can actually have, especially if that next child is also Rh positive. So once again, during the first pregnancy, some of the red blood cells of the fetus that have antigen D Rh factor can leak into the mother's blood. Now, this leakage of red blood cells that contain antigen D into the mother's bloodstream, who is Rh negative, will cause the immune system of the mother to begin producing antigen D antibodies. However, since the fetal red blood cells usually leak during childbirth, this will not affect that fetus. But let's suppose the same woman who has been exposed to these antigen D proteins and now contains those antibodies against antigen D, let's suppose she once again becomes pregnant with a fetus who is once again Rh positive. What will happen now? Well, now we're going to have something called Rh incompatibility. And what that means is because these antibodies that were produced as a result of that first pregnancy are floating around in the bloodstream of the mother, and because they're small enough, they can pass across the fetal membrane into the bloodstream of that fetus. Now, how is this a problem? Well, the fetus is assumed to be Rh positive, and that means it contains red blood cells that contain the antigen D. Now, when these antibodies travel through the membrane of the placenta and into the bloodstream of the fetus, they will bind onto the antigen D proteins of the red blood cells of the fetus. And once bound, they will lyse the cell. And when the cell breaks, when it lyses, it releases dangerous chemicals into the bloodstream of that fetus, and that can ultimately damage the organs of that fetus, including the brain and other organs. So we can see how, as a result of our immune system trying to protect our body, it can actually damage that fetus during the process of pregnancy second time around. So once again, to conclude, antigen A and antigen B, the antigens that determine our blood group, are not the only antigens that can be found on the membrane of red blood cells. Another type of antigen is antigen D, and this is part of a group of antigens found in humans known as the Rh factor. Our immune system displays something known as immunological tolerance. So our white blood cells of our immune system are naturally immunologically tolerant to the healthy cells of our body. And what that means is these white blood cells will not attack the healthy cells of our body. Why is that? Well, because our healthy cells produce proteins called self-antigens, and they display these self-antigens on special protein complexes found on the membrane of those healthy cells. And they display these antigens to white blood cells. So when a white blood cell sees a self-antigen on a healthy cell, it will not attack that healthy cell. So under normal and healthy conditions, our immune system has no problem distinguishing between these self-antigens found on healthy cells and pathogenic antigens found on infected cells or on invading pathogens, such as bacterial cells. However, in certain cases and in certain individuals, our immune system can actually lose its natural immunological tolerance to the healthy cells of our body. And what that means is our immune system can no longer distinguish or differentiate between self-antigens of healthy cells and pathogenic antigens. And at this point, the white blood cells will begin to attack not only the pathogens and infected cells, but also the healthy cells of our body. And this condition, in which our immune system loses its natural immunological tolerance to our healthy cells, is known as autoimmunity or autoimmune disease. Now, what are some examples of autoimmune diseases? Well, multiple sclerosis is one, rheumatoid arthritis is a second one, diabetes type 1 is a third one, and we also have myasthenia gravis, which we'll focus on briefly in this lecture. So what exactly is myasthenia gravis? Well, this is an autoimmune disease of our body that affects our skeletal tissue. So basically, our body begins to produce an antibody that it normally shouldn't. And when this antibody is produced, it begins to circulate inside our blood and it binds onto acetylcholine receptors